Uh, Hello, everyone. My name is Ashish Kokkula. I'm a second-year biomedical engineering student here at NJIT, and today I'll be talking about the mechanism of action of platinum nanoparticles in an aggressive form of breast cancer called triple-negative breast cancer. So to start with a little bit of background, triple-negative breast cancer is a very aggressive form of breast cancer. It lacks 3 common hormone receptors found in regular breast cells and other types of breast cancer cells. Specifically, these receptors are estrogen, progesterone, and human epidermal growth factor. What makes this cancer so unique is the fact that since it lacks these 3 receptors, it's very difficult for common chemotherapies to actually target and kill this type of cancer cell. Most current chemotherapies use this use these receptors as targeting mechanisms. But when when these receptors are not there, it's very hard to actually target and kill the cancer. So I want to go into a little bit about platinum nanoparticles. So platinum nanoparticles are platinum particles at the nanoscale, 10 to the negative 9 meters, and they've been shown in various different research already that they have potential anti-cancer activities. They've been used in tested against liver cancer, colon cancer, and other types of breast cancer. And the main thing about platinum nanoparticles is the fact that it has low toxicity towards healthy cells. And in chemotherapies and cancer treatments, that is a golden property to have, to be able to be unique and to have low toxicity towards healthy cells while being toxic towards cancer cells. So some current platinum-based treatments that are out there in the market right now are the main one being cisplatin, and then derivatives of cisplatin, carboplatin and oxaliplatin. These platinum-based drugs are very effective against cancer, but they are highly toxic towards healthy cells. So the main thing with cisplatin, the way that it works, it essentially enters a cell, ionizes within the endosomes of the cell to become platinum 2+ ions, and then these platinum 2+ ions enter the nucleus of the cell and bind to the DNA of a cancer cell. The platinum ions break the DNA and essentially inhibit the cell's ability to replicate and proliferate. So how is this involved in my project? So we need to understand why platinum nanoparticles actually work against triple-negative breast cancer and other types of cancer cells. Because if we understand the why and the how, we can understand why certain patients are not responding well to this treatment, whereas other patients might be responding well to it. So what my proposed mechanism is that the platinum nanoparticles work similar to the cisplatin mechanism. We're using cisplatin as a base model and then building off of that model. Another component to the mechanism is that the replication speed of the platinum nanoparticles, of the cancer cells, affects the platinum nanoparticles' efficacy. Additionally, the ROS production, the amount of reactive oxidative species that are produced when platinum nanoparticles are uptaken by cancer cells, is not involved in the mechanism. And finally, the lower pH environments increases platinum ion concentrations, which would kill the cancer cells a lot more than if you were in a healthy pH 7 environment. So to start, I conducted some cell viability tests using 2 different cell lines, triple-positive breast cancer and uterine sarcoma. So triple-positive breast cancer, it's the almost exact opposite of triple-negative breast cancer. It tests positive for the 3 hormone receptors, estrogen, progesterone, and human epidermal growth factor. And the main thing about this cell line is that it replicates slower than triple-negative breast cancer, but faster than healthy cells. The uterine sarcoma there's a slow prognosis for this type of cell line, but the specific cell line that we bought for uterine sarcoma is a cisplatin-resistant cell line. So the reason why we're using this cisplatin-resistant uterine sarcoma is because if we're proposing that the platinum nanoparticles are similar to the cisplatin mechanism, then that means the uterine sarcoma should not be affected by the platinum nanoparticles at all. And to conduct these cell viability tests, I did some MTS assays. MTS is a solution that, when uptaken by live cells, the live cells metabolize the MTS into formazine, and then we put them through a plate reader that excites the cells at 495 nanometers, and then emits a wavelength back that we can use to quantify our how many cells are actually alive. So to go into some of my results for cell viabilities. These cell viability tests are used, and we're using only bare platinum nanoparticles. And with the triple positive breast cancer, we can see that there is a decrease in triple positive breast cancer cells at high concentrations over a longer period of time. However, the triple positive breast cancer is not affected by the platinum nanoparticles as much as the triple negative breast cancer cells that we see, and I'll go back for this, as we see for the triple negative breast cancer cells. The triple negative breast cancer cells are much more affected than the triple positive breast cancer cells. And this supports my hypothesis that the triple positive breast cancer, which replicates slower than triple negative breast cancer, it supports the hypothesis that replication speed is a factor in the mechanism of platinum nanoparticles. Now, to get into uterine sarcoma, being that it is a resistance to cisplatin, the uterine sarcoma is affected by the platinum nanoparticles in the first day. However, we see this in all the different cell lines that we have tested platinum nanoparticles against, whether it is this triple positive or triple negative breast cancer or healthy fibroblasts. However, the key point here is the fact that the uterine sarcoma recovers by day 5, even at high concentrations of platinum nanoparticles. Almost 95% of them do recover, which means that the platinum nanoparticles must follow a similar mechanism to the cisplatin mechanism. Now, what I'm currently working on is really pinpointing that mechanism. And I've set up the 3 different types of experiments, and the first one being reactive oxidative species production experiments. So reactive oxidative species are highly reactive compounds that are formed when cells uptake any certain nanoparticles or any other types of drugs, and you can see them when you see hydrogen peroxide that is produced outside of the cells. Current literature claims that these ROS species are what causes platinum nanoparticles and other drugs to really kill a cancer cell. However, all cells produce ROS whenever they uptake in nanoparticles, and some cells still survive after taking up nanoparticles and producing ROS. So there must be some other reason why platinum nanoparticles kill triple negative breast cancer. So I'm proposing that the ROS is not involved in the main mechanism of platinum nanoparticles. So I have some results for the triple negative breast cancer, and we see there is a significant decrease in ROS production when bare platinum nanoparticles are administered to triple negative breast cancer cells. And this is very key because it supports my hypothesis that there is ROS is not involved in the mechanism of platinum nanoparticles in triple negative breast cancer. But of course, I'm also testing this against 3 other cell lines: uterine sarcoma, triple positive breast cancer, and healthy fibroblasts, to see if there's a similar trend. And some preliminary results, especially with the uterine sarcoma, shows the same amount of the same thing. We see a lot of variability in the first day, but it's really the 5 days that we're particularly interested in. The ROS does not go above 100%. It does not make more ROS than our control group of when we don't administer any platinum nanoparticles at all. And that is what is key. That is what we're trying, that we want to see. And I see at the current moment, preliminary results for triple positive breast cancer are showing similar trends. Now, research is still very much ongoing, and I am still working on my other 2 experiments, which are platinum ion release studies and nanoparticle uptake studies. So the platinum ion release studies, I'm setting up 2 different setups where basically there's a pH 5 setup and a pH 7 setup. And the pH 5 is to replicate the cancer cell environment, and the pH 7 is to replicate the healthy cell environment. And this is essentially to show whether a lower pH environment is involved in the mechanism for platinum nanoparticles. The idea there is that if there's more in an acidic environment, platinums will ionize a lot more frequently, and the cancer cells will uptake the platinum ions much more frequently with the platinum ions binding to the DNA and breaking the DNA of the cells and killing the cancer cells. Whereas in a healthy cell environment, this would not be the case. And now the nanoparticle uptake is really just to confirm that regardless of cell type, all cells will uptake the similar amounts of nanoparticles, regardless of whether it's a fibroblast, triple-positive breast cancer, uterine sarcoma, etc. So I'd like to acknowledge my professor, Dr. Kathleen McKennis, and the PhD student that I'm working with, Ida Lopez-Ruiz, for helping me and advising me through this project, as well as the new NJIT Undergraduate Research Innovation and the New Jersey Health Foundation for funding my project.
This is like, I'm far from my base here, but you're saying that ROS is not important
Yes.
and something else important, and what I understand you're talking about is ionization of platinum?
So the ROS is they're just compounds that are produced when cells uptake any sort of particles. Now, the platinum ions are completely separate from that, but what I'm basically saying with the ROS is that there's current literature that's saying that ROS is what is causing cells to die.
I'm just trying to, so you're talking about platinum ions, but you have platinum nanoparticles.
Yeah.
So where are the ions coming?
So the ions are coming, if I can go back really quickly. The ions come from the fact that they ionize within a cell. So outside of the cell, they are neutral, platinum, but once they enter the cell, in endosomes have a lower pH. They are normally at a pH 5, and in those endosomes, the platinum ionizes into these 2+ platinum.
Thanks. Can I ask a maybe similar follow-up?
Yeah.
So ROS is always going to be produced when nanoparticles are being uptaken, right?
Yes.
So in your kind of last experiments, maybe I missed it, but how did you actually disentangle the effect of ROS being produced to the fact that it's not important in the cell degradation?
So essentially, it's that we're seeing that ROS is not being as much. There's not as much ROS production over long periods of time after platinum nanoparticles are administered. If we were following current literature, we would see an increase in ROS production over a longer period of time, rather than what we're seeing now, there's being a decrease in ROS production, which we can equate to that being that, ROS is not being involved in the mechanism at all.
Okay. Oh, yeah.
A different question. You showed a bunch of plots like this, and there's like a bunch of bars and then a straight line.
Yeah,
how does a straight line, how do you make up that straight line? Is there a process to it, or just like an eyeballing?
So I did conduct some I did conduct some significance tests on these studies here. The red line is really to show just a trend that we've been seeing throughout for different, whether it's a cell viability test and for the ROS production test. It's just to see what is the trend for different concentrations of platinum, for different concentration of platinum nanoparticles, and how is it
different days show different trends, it looks like.
Yeah.
But I doesn't know. It's like
so the red line is really more for the red lines are more for an eyeballing. It's just for anyone who's just looking at it at the graphs. But essentially, we're just seeing generally across, especially for the ROS production, there's a decrease in ROS production regardless of cell line.